Hi everyone, I am Sanjana. I'm a second year PhD student at the University of Edinburgh. And today I'll be discussing a recent study we started working on, looking at the association between telomere length and immunity in a natural soy sheep population. So as evolutionary ecologists in the field of aging, we're broadly interested in understanding what drives variation in aging in the wild. And we can look to proximate mechanisms and physiological processes that contribute to senescence. The recent interest has been the study of telomeres. So telomeres are repetitive DNA sequences present at chromosomal ends. And there has been some support for telomere length declining with age across different species. It's also been proposed as a potential marker of individual physiological state, as well as playing a role in mediating trade-offs between different life history traits. Another physiological process contributing to senescence could be variation in immunity and resistance to parasites. So in many populations, exposure to parasites and immune resistance impacts survival and lifespan. And there has also been evidence for decline in immune function with age, also known as immunosenescence in wild populations. So along with understanding how these processes individually contribute to senescence, I think it's also interesting to map out how these processes themselves are linked. So in terms of the association between telomere length and immunity and parasitism, there have been two causal mechanisms that have been proposed. So one idea is the cost of infection hypothesis, where becoming infected and investing in an immune response comes at a cost to somatic maintenance that manifests as a decrease in telomere length. Or you could also have it the other way around, where aging and telomere shortening as a result of aging can lead to decline in immune function or immunosenescence. So there have been studies that have explored this association, and experimental studies in lab animal models have shown support for the cost of infection hypotheses. In terms of what this relationship looks like in the wild, there have also been studies done in some birds and mammal populations looking at the impact of microparasitic infection on telomere length, and experimental studies in the wild as well showing support for the cost of infection hypotheses, while there have been a couple of correlational studies done as well with mixed support. So overall, there is limited evidence available for understanding the impact of different types of infection on immune cell telomere length, and the potential costs of mounting an immune response on telomere length is also largely unexplored. We also don't know whether telomeres act as a mediator of trade-offs between life history traits like immunity and survival. So we explored these questions in the soy sheep. So the soy sheep on St. Kilda are part of an intensive study since 1985. We have extensive data about their life history and phenotypic data available as well. So in August every year in the summer, we go to these islands and we catch the sheep, and we also collect blood and fecal samples. So in terms of the parasitism and immunity in this system, the soy sheep are chronically infected with gastrointestinal macroparasitic nematodes, with the most prevalent species being the strong-guile nematodes. And the fecal egg counts serve as a good proxy for parasite burdens in this species. We also have information about IgG antibody levels in their blood, and this is a good marker of protective immunity and resistance to parasites in this system. There has also been previous published work that has looked at the relationship of these parasitic and immune measures and how they relate to age and survival. So we find fecal egg counts to increase with age and be negatively associated with survival, while IgG antibody levels decline with age and are positively associated with overwinter survival. We also have information about their telomere length, and this was estimated from their white blood cells using qPCR assays. And we measured their relative telomere length, where telomere length is measured relative to a control or calibrator sample that's run on each of the qPCR plates. Again, previous published work showing telomere length to be weakly declining with age and positively associated with overwinter survival. So in our study, we had about 2,700 observations collected from around 1,300 individuals between 1998 and 2015. So our first question was in understanding whether there's an association between telomere length and parasitism and immunity in the soy sheep, and also whether these associations are being driven by differences among individuals due to their genetics, perhaps, or whether it's within individual variation that is driving any associations that we may observe. So to understand the impact of parasitism on telomere length was modeled as a Gaussian response variable and included fixed effects of strong fecal egg counts and age. And to understand the impact of immune response on telomere length, we included a fixed effect of the IgG antibody levels. Both these models included ID and year to account for repeated sampling of individuals across different years, as well as qPCR assay-related variation. So what we find is telomere length and strong guile burdens to be positively associated. So you can see on the plot here, the dark line is the median from the posterior distribution, and the lightly colored lines are all 2000 posterior draws from the posterior distribution. And the dots are the raw data. So we find a positive association between strong guile fecal egg count and telomere length. And we found no association between telomere length and IgG antibody levels. We also estimated between and within individual effects in the adults. So we ran a trivariate model that included telomere length, fecal egg count, and IgG levels as response variables. We accounted for the relevant fixed effects as well as random effects of year and assay-related variation. But importantly, we also modeled the variance due to differences among individuals, which is captured in the ID random effect, as well as the residual variance, which reflects the within individual variation. What we found is a positive within individual correlation between telomere length and strong guile fecal egg count. So you can see on the plot here on the left, a positive correlation at the residual level is what is driving the positive association we previously observed between telomere length and strong guile fecal egg count. The plot on the right here depicts the individual repeatabilities for telomere length and strong guile fecal egg count, as well as IgG antibody levels. So we see here that almost 25% of the variation in telomere length and strong guile fecal egg count is due to differences among individuals, while a lot of the variation is captured in the residual component. We were also interested in understanding whether telomeres mediate the relationship between parasitism and overwinter survival. And so we can do that by running a path analysis using multivariate models to evaluate specific causal graph diagrams. So over here on the left, the direct effect of parasitism on survival can be estimated, as well as the indirect effect mediated through telomeres. So these models as well accounted for fixed effects of age and sex and random effects of individual year and assay-related variation. So we find that telomeres do not mediate association between parasitism and survival. So here you can see the direct effect of parasitism and immunity on survival. So having high antibody levels and low parasite burdens mean higher survival probability. But when we look at whether this relationship is mediated by telomeres, we find that no support for this. So to conclude and summarize the preliminary results, we find telomeres and strong guile burdens to be positively correlated at the within individual level in adults. And this is also independent of any protective immunity effects that are conferred by IgG antibodies. So essentially what this means is that individuals with lower parasite burdens in a given year compared to the average across their life also have shorter telomeres that year. This suggests that recent physiological variation, either investment reproduction or short-term environment-related effects, could be underlying the positive association observed. We also found no association between telomere length and IgG antibody immune response in this system, as well as the path analysis revealing no support for telomere as a mediator of trade-offs between immunity and survival. This suggests that in this system, telomeres are a context-specific marker and captures some aspects of among and within individual variation. The previous work has shown telomere length to be moderately linked to survival at the among individual level and weakly linked at the within individual level to some aspects of maternal reproduction and parasitism, and also not associated here with immunity or parasite resistance. So with that, I'd just like to thank everyone involved in the study, as well as all of the people who worked in the lab to collect all of this amazing data, and the people involved in the long-term project as well. And thank you all for listening.